my name is Dia.
Hi, my name is Nur Qadirah.
What do you think about that? Hi, my name is Adriana.
Hi, my name is Alya.
Guys, where is Adriana? Yeah, where is she?
She late again.
She always let us in this kind of situation.
Hi, my name is Aiman.
Sorry guys, I'm late.
Yes, I'm sorry.
That's all for chapter 6, student. Next week we will study a new chapter for recombination of DNA technology. So I want you guys to prepare a presentation about this chapter along with your creativity. Make sure you're prepared. Thank you.
Thank you, sir. Bye.
Thank you,
Thank
sir.
you, sir.
Bye.
Guys.
Oh, what should we do for our group assignment? I have an idea.
So, the definition of recombinant DNA of technology is a method in which the DNA molecules make in vitro by combining the gene from different organisms.
There's 5 tools used in recombinant DNA technology. The first tool is target DNA. What is it? Target DNA is a sequence of DNA containing gene of interest. Gene cloning and genetic engineering generally rely on the use of enzymes that cut DNA molecules at a limited number of specific locations. These enzymes called restriction enzymes or restriction endonucleases. What is it? Restriction enzyme is enzyme which cuts DNA molecules at a restriction site and produces short restriction fragments. Restriction site is the short base pair sequence. So it consists of 4 to 8 base pair recognized by a particular restriction enzyme. Restriction site contain palindromic where the same sequence of bases is found on both DNA strands but in opposite direction. There are 2 examples of restriction enzyme. The first one is EcoR1. EcoR1 produced by E. coli. So it recognizes the base sequence of 5' GAA TTC 3'. So it will make staggered cuts within the sequence. So GAA TTC, it will cut from G. And then it will produces sticky end, single-stranded end, like this. So the second example of restriction enzyme is MAR1. It produced by Serratia marcescens. It recognizes the base sequence of 5' CCCGGG 3'. The cut is made between the adjacent C and G, like this one, and it will make straight cut and produces blunt ends. The third tool is DNA cloning vector. A DNA cloning vector is in plasmid. So, DNA molecule which can carry foreign DNA fragment into a cell and replicate in that cell. Its characteristics: able to accept foreign DNA in MCS, multiple cloning sites. And the second one, able to replicate freely in the host cell, origin of replication, initiation, or ORI. So the third one, possess selectable genetic marker. So it will resistant to antibiotics. So the example of cloning vector is plasmid.
Okay, in order to replicate, DNA vector that carry target gene is introduced into the host cell. So, this is another meaning of it. A cell which receives a recombinant DNA and allows the replication of recombinant DNA.
the gene products from your recombinant DNA. So bacteria is the most commonly used as host cell. Reasons: because DNA can be easily isolated from or introduced into a bacterium. Bacterial cultures grow quickly, rapidly, replication any foreign genes that carry. Example types of bacteria is E. coli. So the last tools is modified enzyme, which is also known as DNA ligase. DNA ligase, the function of it is to join the foreign DNA fragments containing gene of interest and bacterial plasmid. Another word is it joins the sections. And then by catalyzing the formation of a polyester bond, which is also known as covalent bonds, to form recombinant DNA. Okay, for that polymerize, it is used in polymerase chain reaction, which is also known as PCR. And it's another method of applying short segments of DNA. And it also replaces the DNA polymerase because of the temperature condition of the PCR. And also to add nucleotides to the 3' end of primer in order to produce new DNA strand.
This is the plasmid that isolated from the bacteria cell. And then this is the registration site, which the registration enzyme will tag the registration site at the plasmid. This is gene of interest, which will join the plasmid by DNA ligase to form a recombinant DNA. So I want to explain to you guys about the 5th step of gene cloning. So this is the petri dish. This screening process is done using blue-white screening technique. The function of the screening process is to identify bacteria that carries the recombinant DNA. So at this petri dish contains 2 colors of bacteria, which are blue and white colonies. So which color should we choose in the end of the process of this screening? The answer is white. Okay, so for the step in gene cloning, the first step is isolation of gene. Okay, so plasmid from bacteria cell is isolated. Okay, so this is it. Okay, and then gene of interest is isolated from DNA of donor cell. And then this.
Okay, the next step is cleave and cut. Okay, DNA donor from human and plasmid bacterial is cleaved using a compatible restriction enzyme, which is DNA ligase. It will make a single cut within the laxi gene site of cleaving. It will produce a complementary sticky end or blunt end.
Okay, so for the third step, insertion. DNA fragment, which is gene of interest, is inserted into the plasmid. Okay, so it inserts into the plasmid, and then it joined together by DNA ligase to form recombinant DNA. As you can see, it inserts into it and then it forms recombinant DNA plasmid.
The fourth step is transformation and amplification. The recombinant DNA are then inserted into the bacteria through the transformation process. Next, for the amplification, as the bacteria reproduce, the recombinant of DNA will be replicate or be cloned and produce much more the recombinant of DNA like this. Okay, for the last step is screening. Screening process is done by using blue-white screening technique.
Okay, so for the first step is plate the bacteria in agar that contain antibiotic ampicillin and agar.
From that process, there will be 2 options, which is for the first one is not resistant to antibiotic ampicillin, the bacteria will not grow.
So for the next option is it will be resistant to the antibiotic ampicillin, so that the bacteria will reproduce and form colonies.
Okay, from that process, there will be 2 options. First one is colonies without the recombinant plasmid will be blue. Why it is blue? Because it contains intact laxi. I can produce beta-galactosidase and hydrolyzed agar.
And then, if the colonies with recombinant plasmid, it will be white. Why it is white? It is because it do not contain intact like Z, and then it cannot produce beta galactosidase, and the last one is it cannot hydrolyze X-gal.
Thank you very much, Madam Rudziah, our biology lecturer, for teaching us.
And thank you to our teammates because give a well cooperation to this task.
And not to forget to our classmates, thank you for helping us to finish this project.
Thank you to our parents for giving support to us and encourage us to finish this assignment.
Bye!
Bye!